The acid-fast stain is another differential staining technique used to distinguish organisms with a waxy outer layer. The layer, mycolic acid, prevents other staining procedures from working by not allowing for the decolorization following a wash with acid alcohol. The genus that this is used on is Mycobacterium. For you to remember easily, Mycobacterium contains mycolic acid, myco. Today we'll be using a primary stain, carbofusion, decolorized with acid alcohol, and counterstain with methylene blue. You will follow the same procedure for your bacterial smear preparation. It is recommended to cover the specimen with tissue paper, but it can also be completed without this. You'll be using the steaming technique in order to allow for the primary stain to penetrate the cell. To conduct the steaming technique, simply place the stain on your specimen. Next, take your slide carefully to the flame and slowly pass it through the flame. Continue to do this until you begin to see steam coming off the slide. Once the steam has stopped, continue passing it through the flame until it begins again. This steaming procedure goes on for 5 minutes, upon which you will remove the tissue paper, if used, and drain and wash with DI water. Care should be taken to not allow for the stain to dry fully on the slide. Additional stain should be added when this is the case. The next step is to decolorize with acid alcohol. Non-acid fast bacteria will lose the primary stain, but the Mycobacterium will retain it. Rinse with DI water and counterstain with methylene blue for 1 minute. Lastly, wash with DI water and drain. You do not use bibulous paper on this slide, as you may remove the organism. Allow the slide to air dry. When visualizing the mixture, you will see that acid-fast Mycobacterium will stain red, while non-acid-fast Staphylococcus will pick up the counterstain and be blue.